for your attention, and I'd be happy to answer any questions as Dr. Fletcher sees fit.
So I would like to suggest, in the interest of time, because I think it'll work well, why don't we take questions after Dr. Butrinsky's talk, if the two of you can field questions together.
Cancer Research Foundation for all of the support for this group, as well as many groups that they support. It's also great to see our patients in the audience, both GIST and non-GIST patients. I'm going to carry on as to what Dr. George started, but I would also like to step back, and I'm going to break my talk into two sections. One is just to discuss the nuts and bolts of clinical trials, because I think there are some people in the audience that might not be on a clinical trial. I'd also like to then jump into some of the new, exciting, targeted therapies that are being used in our clinics in hopes to be the next generation of drugs that will work when imatinib and sunitinib and other medications may no longer work. First, the sort of clinical trials 101. What is the purpose of a clinical trial? Essentially, this is a research study involving people. And there are several types of clinical trials. We're focusing mostly on therapeutic clinical trials, but there are prevention clinical trials. We occasionally have maintenance clinical trials. Many of the clinical trials that we participate in also are looking at QOL, or quality of life endpoints, to make sure that in addition to trying to find therapies that are ultimately going to benefit people by hopefully improving how long they survive, we also want to have therapies that aren't causing too much toxicity. Why are we doing clinical trials? I think simply we all want to find the cure to GIST, non-GIST sarcomas, or at least better ways to treat people so they can live longer with good quality of life. When does one participate in clinical trials? Well, I'll show in a later slide, we have clinical trials that really cover the entire spectrum of when people, from diagnosis to periods of time where they've had FDA-approved drugs like Dr. George had mentioned, and require other medications to hopefully allow a good quality of life and a longer existence. Clinical trials are traditionally divided into four main phases. Phase 1 clinical trials are drugs that people have great ideas. They generate a clinical trial document that I'll talk about briefly. Ultimately, bring these drugs from preclinical testing into the clinic, so they're first in human trials. The goal of those trials are essentially to find the right dose. There's often staggered enrollment. There's often start and stops of the clinical trials to allow a certain number of people that are on a particular dose to have received the drug before we put on more people. So it's in an effort to find a safe dose. Although the goal is to find a safe dose, all of us who treat people with GIST and non-GIST sarcomas really want to see these drugs work. And so we're also actively monitoring people with CAT scans or with tumor measurements, et cetera, to see if the drug is actually working based on those evaluation criteria. The phase 2 clinical trial is right on the foot of the footsteps of phase 1 clinical trials. We find the right dose. We hopefully see a endpoint that is achieved, such as a response in somebody with GIST or response in somebody with another type of sarcoma. And then we design the clinical trial to hopefully incorporate a more narrow group of people that may just have GIST or just may have a certain specific subtype of sarcoma that seems like this drug could have some effectiveness. We have the right dose, and so we try to put on somewhere between 30 to 50 people with that particular subtype of sarcoma or GIST, and we ultimately look to see if the drug is effective. So that's the end point. And then if one drug crosses that particular hurdle through phase 2 testing, we bring that drug onto phase 3 testing. That testing is usually incorporating this new kid on the block compared to the older standard. So regorafenib versus placebo is one way to conduct a phase 3 trial. Some clinical trials are comparing imatinib to another drug to kind of compare head-to-head. And that gives us an idea of how drugs work in a head-to-head fashion. And many of those trials, I have in parentheses randomized, many of those trials randomize people into either one arm or the other. There are occasionally, as I will discuss and as Dr. George discussed, placebos or sugar-coated tablets that don't, in theory, have any effectiveness in the particular cancer that we're treating, are used in some trials, those are discussed extensively before anybody participates. People aren't surprised, or they shouldn't be surprised that there's a placebo in a clinical trial that we have discussions about. Phase 4 trials I'm not going to mention, but are more post-marketing trials where we continue to follow over time and over the life of the drug whether this drug has some late-term side effect 10 years, 15 years down the road, and those come out in those sort of trials. The clinical trial protocol document is actually scientists like Jonathan Fletcher and his people in his lab meet with the clinicians and the investigators in our group and discuss better ways to use drugs that are in the pipeline, so to speak, and that ultimately generates a document that says this has promise, let's bring this into human testing. That document goes through intense scrutiny both at the level of scientific review committees as well as ethics committees, which incorporate lay people and people with the disease sometime and scientists, to make sure this makes sense. So 3 months, 6 months, sometimes that can take a year in the making before a clinical trial actually is opened at a particular site. So that's sort of the journey of the clinical trial. The most important topic or most important aspect, I believe, is the informed consent discussion. That's where the clinician is in the room with the potential participant, goes through all the aspects of this clinical trial. That person hopefully had time to review the consent document. And that's where one signs on the dotted line, so to speak, to participate in a trial. It's very clear that participants this is a voluntary decision. One can withdraw at any time. And so that discussion is very important, and that discussion continues during the time that the participant is on a clinical trial. There are clearly benefits and risks to participation. This whole discussion I'm having in 2 minutes is probably hours worth of discussion, but the benefits to participation in my mind is this does increase options people have in situations where maybe their options seem to be a little more limited. These offer other ways to target different aspects of the tumor biology. This offers the possibility that there might be a response or shrinkage of the tumor. There are clearly risks to participation. Sometimes there are side effects. Sometimes the medications don't work the way we had hoped they'd work. So that discussion is had with the clinician in the room, and decisions are made ongoing during the trial. There are clearly barriers to participation. We, as the clinicians in the office, would really encourage people to consider clinical trials and are always disappointed that the numbers of adults participating in clinical trials are less than we would desire. But hopefully over time that we can overcome some of these barriers, which clearly can be logistics. We see people from California. It's tough to get to Boston every week. We understand that. We try to encourage and also actively help people try to find clinical trials that may be closer to their home of origin. There clearly fear for clinical trials. It's definitely an unknown venture, whether you're missing an opportunity to try something else. So these are all discussed, and this process isn't necessarily something that takes 1 or 2 visits, but can take multiple visits. I'm just going to stop there in this brief review of clinical trials and discuss some of the agents that are available at our site, particularly phase 1 agents. This slide just shows, I'm just with this cartoon showing that the GIST cancer cell or the non-GIST cancer cell is composed of proteins that occupy the outer surface of the cells. These are proteins that we've discussed briefly. c-KIT protein sits there. PDGFR alpha sits there. In the middle, where there's this arrow, there are multiple proteins: PI3 kinase proteins, HSP90 proteins, mTOR proteins. These will become more relevant in a moment. All of these proteins seem to play some role in how the tumor cell lives and survives. And our role is to try to find medications, therapies that somehow stop that program so that people can either live with some tumor burden, or if we can possibly shrink the tumor, to live with less tumor burden. Currently, we have 4 main pathways that we're trying to target at the Dana-Farber, and these are also available at other sites. PI3 kinase inhibitors, we have 2 particular PI3 kinase inhibitors that we have on clinical trial. PI3 kinase mTOR inhibitors. This is a medication that inhibits both of those proteins. We also have an inhibitor of a kinase of 3 kinases, c-KIT, FMS, and FLT3. We also have inhibitors of HSP90 proteins. All of these trials that I mentioned are focused not only on for people that have GIST tumors that meet the appropriate requirements, usually having failed imatinib or sunitinib, but also for non-GIST sarcomas. GIST has been really a canary in a coal mine, so to speak, for many of the other sarcomas, and so the lessons that we've learned from GIST we're also applying to many other types of sarcomas. Currently, within the next 6 months, we should have a couple of other clinical trials that will be available at our site that are looking at this concept of combinatorial therapy using an HSP90 inhibitor with imatinib to hopefully get possibly the power of both of these agents, and another medication that's going to be in early testing as an antibody that targets the PDGFR alpha receptor, which we also hope will be beneficial for people that have GIST that is refractory to other therapies. There are a couple of other ongoing trials that we have that are not therapeutic trials. I think Dr. Judy Garber is going to be discussing Project FLAG later, and I won't dwell on that, but we're also interested in the genetics of GIST. We also, within our center, have a clinical database for people that have sarcoma, whether non-GIST sarcomas or sarcoma, that we encourage participation so we can advance the field, hopefully even further. In my summary slide here, I just wanted to show we have trials that cover the continuum. Last year, I used this slide, and even in 1 year, things have changed in the sense of what trials are available. For newly diagnosed localized GIST, patients that have localized GIST that have been resected, we had an ongoing trial looking at imatinib for 5 years. That has recently just closed because it has hit its accrual goals. That data will be available probably in 3 to 5 years to have a sense of how that data looks. Newly diagnosed patients who have metastatic GIST, we were testing imatinib versus nilotinib with many other sites. That trial has recently closed due to superiority of imatinib. And so just in 1 year time, there's been some change in a couple of the larger trials. People that have had imatinib or sunitinib, this is focusing just on people with GIST, we have a variety of clinical trials. Dr. George talked about our priority trial, the regorafenib versus placebo trial, and we have many other phase 1 trials when people aren't eligible for that, or frankly when some people don't want to even consider a placebo-controlled trial. We also see many people that have failed imatinib, sunitinib, sorafenib, and even other medications for GIST, where we have, again, other trials in our phase 1 pipeline that I mentioned earlier. I'd just like to thank the patients, of course, and my colleagues, and I'm here to answer any questions with, I guess, Dr. George.
So we're going to go next with Dr. Janeway with the overview of wild-type and pediatric GIST, followed by Dr. Judy Garber, who is leading a national, I guess international, certainly groundbreaking effort to pin down the genetic predisposition to GIST, which is relevant at least for a small number of GIST patients, and to update us on that effort. Katie?
Thanks, Jonathan, and thanks to everybody for coming and for your attention. So I'm going to tell you about advances in pediatric and wild-type GIST. Just an overview of my talk. Jonathan already told you the definition of what I'm speaking about, which is wild-type GIST. And that really means GISTs that don't have mutations in the KIT or the PDGF receptor alpha genes within the tumor. And the wild-type GISTs are really broken down into 4 different groups. So I'm going to go over each of the different types of wild-type GIST. So there's carny triad, carny stratitis dyad, pediatric GIST, and wild-type GIST. And that wild-type really means the wild-type in the adult. As I go through the talk, the things that are listed in blue are really recent discoveries in the last 2 or 3 years. And this is an area where we've had a lot of recent discovery, which is really great because this type of GIST is something that we know a lot less about than the sort of standard type of GIST with the KIT or PDGF receptor alpha mutations. I'm going to tell you about the natural history and treatment of these types of tumor GIST, and then about research efforts.